Hello everyone, welcome back. We'll be continuing with the metabolism of amino acids. In the previous presentation, we concluded with the types of deamination, that is oxidative deamination, reductive deamination, hydrolytic or non-oxidative deamination, and intramolecular deamination. In this presentation, we'll be looking in detail the oxidative deamination. So what is oxidative deamination? Deamination is removal of ammonia, free ammonia, and oxidative because the whole reaction is coupled with oxidation. So oxidative deamination, it is the oxidative removal of the amino group from amino acids. Okay, here the whole reaction, it is coupled with oxidation. So it is almost, we can say it is a redox reaction. Okay, and this process, it is very important when an organism has more nitrogen in the nutrient than what is required. Okay, and here you can see since ammonia is released, it becomes a source for urea synthesis. And apart from ammonia, the amino acid, the main carbon skeleton of amino acid, it gets converted to keto acid, the alpha keto acid, and the alpha keto acid can enter various other reactions. The whole oxidative deamination mainly occurs in liver and kidney because the enzymes required are present in these organs. The process, it is stereospecific. It is catalyzed by L and D amino acid oxidases. So these enzymes are stereospecific in the sense L amino acid oxidases, they act upon L amino acids, and D amino acid oxidases, it acts upon D amino acid. They are stereospecific. The first step we can see, it is the removal of two hydrogen atoms by the flavin coenzyme with the formation of an unstable intermediary alpha amino acid intermediate. Okay, this intermediate undergoes decomposition by addition of water and forms the ammonium ion and corresponding alpha keto acid. Okay, we can see over here. So this is the whole process what was explained. That is, it splits an amino acid. The whole the process, it will split amino acid to ammonia and the corresponding alpha keto acid. The whole reaction is catalyzed by the oxidases enzyme, amino acid oxidases, or we can say amino acid dehydrogenases. And here the oxidation, it is carried out with oxidizing agent, that is a flavin, usually flavin coenzymes. Okay, it could be FMN or FAD. Okay, so with the help of this coenzyme, the enzyme oxidases, it will help in splitting the alpha amino acid to the respective keto acid and ammonia. Now, as already mentioned, it takes place in two steps. The first step is oxidation and the second step is hydrolysis. In the first step, what happens is the alpha amino acid, it undergoes dehydrogenation with the oxidizing agent flavin and the enzyme L-amino acid oxidases. Okay, the FMN or FAD, it becomes oxidizing agent which receives the hydrogen which is removed from the alpha amino acid. So here you can see the hydrogen over here and the hydrogen from the ammonia, it is being released, and as a result, the resultant product is a amino acid, C double bond N, isn't it? Amino acid, alpha amino acid. Okay, so this flavin, it takes up the hydrogen atoms and it gets converted to flavin H. It could be FADH2 from FAD and FMNH2 from FMN. Okay, it gets reduced. Fine. So alpha amino acid, it is an intermediate, it is highly unstable and it undergoes spontaneous hydrolysis. Addition of water, you can see over here. It is spontaneous hydrolytic deamination, that is hydrolysis takes place, at the same time ammonia is released. So this particular enzyme catalyzes the conversion of alpha amino acid to the alpha keto acid. Okay, the what all are the products now here? We have reduced FMN or FAD, reduced FAD or FMN. We have ammonia, we have alpha keto acid. So what will happen to each of these? We can see. Okay, the reduced FADH2 or FMNH2, they get spontaneously reoxidized to form the FMN or FAD by molecular oxygen, with the help of molecular oxygen, which itself get reduced to form hydrogen peroxide. And this particular reaction, it is catalyzed by the catalases, the enzyme. And this catalase, the enzyme, it is present in the peroxisomes and glyoxysomes. Okay, now what happens to ammonia? Ammonia enters the urea cycle. Okay, it is used for the synthesis of urea, and we can see ammonia, it is a toxic product, and in monotelic animals, that is where the animals excrete nitrogenous the waste produced by nitrogenous metabolism as ammonia. In them it is excreted as such, but in others it gets detoxified, forming glutamine or uric acid, etc. Okay, coming to alpha keto acids, it will enter the mainly the Krebs cycle. Okay, we can see in examples later. Now this is D amino acid. Actually, for protein synthesis, it is L amino acid being utilized. While D amino acid, it is not useful for protein synthesis, it is not used up. But when it is subjected to oxidative deamination with the help of the enzyme D amino acid oxidases, it gets converted to alpha keto acid. This alpha keto acid on reamination, that is addition of amine group again during transamination process, which will result in L amino acid. And this L amino acid, it is metabolically active and it can be used for the protein synthesis. Okay, so even conversion of D amino acid to metabolically active L amino acid can take place with the help of the process. Fine. So this is a specific example, glutamate. Okay, glutamate, it undergoes oxidation over here. That is the first step. And then hydrolytic deamination. Okay, so oxidation, it results in the formation of amino group, and then hydrolysis, it will result in alpha ketoglutarate. Okay, I hope you remember alpha ketoglutarate was an intermediate substrate in Krebs cycle. Okay, so this is oxidative deamination. The second one is non-oxidative deamination or hydrolytic deamination. Here the major enzymes which are involved, it is amino acid dehydratase and amino acid desulfhydrase. Amino acid dehydratase, it deaminates amino acids which contain hydroxyl groups. And these enzymes are present in liver, so the whole reaction takes place in the liver. Now, which are the hydroxyl containing amino acids? They are threonine, serine, and homoserine. Okay, and you can see here the first step in this particular reaction, it is the dehydrolysis. Okay, or dehydration, we can, sorry, it's not dehydrolysis, dehydration. Okay, dehydration, which is followed by hydrolysis. Okay, so it is dehydration, water is removed. So serine, it undergoes non-oxidative deamination, results in the formation of an unstable amino acid intermediate product. Okay, and this undergoes hydrolysis to give rise to pyruvic acid. So the first step is hydration, and the second it is followed by the hydrolytic deamination. Okay, and this results in the formation of keto acid and ammonia. The whole process is catalyzed by serine dehydratase. Okay, serine is undergoing the process of serine dehydratase, and the coenzyme required, it is pyridoxal phosphate, or we can say PLP. Okay, the next one is desulfhydrase. So this enzyme, it is present in liver, kidney, and pancreas, and the enzyme, it catalyzes deamination of sulfur containing amino acids like cysteine. Okay, and cysteine, homocysteine, etc. The first step in the whole reaction, it is desulfhydration, that is removal of hydrogen sulfide, H2S, and this is followed by the hydrolytic deamination. Okay, the first step, H2S, removal of H2S, it results in the formation of alpha amino acid from the cysteine. Okay, and the hydrolytic deamination, it will result in the formation of pyruvic acid. Okay, again the desulfhydrases also require pyridoxal phosphate as the coenzyme. Okay, I hope it is clear. So this is the example here. You can see the specific dehydratase. Okay, serine, threonine, and homoserine, it undergoes dehydration with the help of dehydratase, and the enzyme is coenzyme required is pyridoxal phosphate. It will result in the formation of an unstable alpha amino acid. This will undergo hydrolysis and deamination to result in the formation of alpha keto acid. Okay, then this is summary of deamination. Oxidative deamination, the L amino acid will give rise to alpha keto acid and ammonia. Right, first one, it is oxidation, and then second one, it is hydrolytic deamination, the steps involved. Okay, non-oxidative deamination, it is dehydratase and desulfhydrases involved. Okay, so this is, you can see here, oxidative deamination, it involves L amino acid oxidases, D amino acid oxidases, and glutamate dehydrogenase. And non-oxidative deamination, two enzymes, dehydratases and desulfhydrases, are involved in the catalysis. Okay, so this is all about the deamination. Thank you.